Thanks for that introduction. We still haven't had the conversation about whose surname is spelled correctly.
True.
I'm going to talk to you a little bit about my lab's work using iPS cells, specifically to model chemotherapy-induced cardiotoxicity. So part of the reason, part of the advantage of iPS cells is we think we can answer unique questions that otherwise have been inaccessible. And where we see this fit into the grander scheme is currently, if you go to see your clinician, they will take your blood pressure, listen to your heart, put an oxygen monitor on you. And it's very clear to everyone that in the near future, this modality will change. And human genomes now, in my lab, the last human genomes we sent out were $600 each. That's going to be very cheap and affordable. There has been slow progress made with access to electronic health records. Many of us now wear personal monitoring of some form, whether that's just your heart rate or your blood pressure. And of course, we're going to combine all this data together to be able to make large-scale comparisons. So where we see the iPS cells fitting into this model is that in the future, if we have every patient's whole genome and we have their essentially phenotype collected in a manner in which we can query it correctly, we can then take the human iPS cell model and validate the genomic hits that we discover that are associated with these phenotypes. And from that, particularly in the case of chemotherapy-induced cardiotoxicity, we'll be able to develop genome drugs that are, their development is informed by the genome as well as therapy modification, and quite simply just a genetic test to be able to predict which patients are going to have which type of response to this drug, whether it be positive or negative. So the iPS cell model is good at answering a very certain type of question. And in the case of chemotherapy-induced cardiotoxicity, this is a really good example of how we could utilize this tool. So if we have 100 patients, they're all treated with the same drug, and 10 of them have a negative effect, such as cardiotoxicity. Our question is whether we could predict this patient-specific drug response, whether it is deterministic or random, whether we can identify the genetic cause of this drug response, and then finally, by finding that genetic response, what can we use to be able to essentially identify genes, pathways, and then develop drugs to modify that drug response. So, for those that don't know, iPS cells are now very simple to make. In the field, we've had nearly 10 years of developing this tool. Now we can make it from a very small volume of blood. We work primarily with pediatric patients, so we do that from either a 3 milliliter blood draw or a finger prick, which is up to about 500 microliters of blood. We expand a specific blood cell type. We use a non-integrating virus to overexpress 4 genes that transform the cells from a essentially red blood cell progenitor into a pre-iPS cell state. We then grow these out over time, and we've spent a lot of time and energy developing chemically defined and cost-effective methodologies for doing this. The cost in my lab is now very, very cheap because we have invested in making this accessible for ourselves. My lab generates around about 100 iPS cell lines a year, and that is only going to expand. That means we do lots of patient recruitment. We've had to completely change our modalities to be able to do this sort of thing. And the iPS cells we make, this is what they look like. And I think one of the things that's easy to overlook is the fact that these cells are just a human genome captured in cell culture, essentially functionally immortal, and with the capacity to differentiate into any of the 220 or so cell types in the body. So a very, very powerful tool for genomics. And I think it's kind of undersold that the utility of the mouse for studying genetics, I think the major tool is going to be human iPS cells for human genomics. So, as I've mentioned, we like to make cardiomyocytes. We spent a lot of time over the years developing these protocols, right back from the very first time anyone had described a directed cardiac differentiation protocol. Before that, they were all just random, achieving about 23% cardiomyocytes. Expanding that over time, we published the first high-efficiency differentiation, and then a few years ago, the first chemically defined, so growth factor-free, completely small molecule, fully known component differentiation. This system now is very, very simple. We have a media that has just 3 components in. That's very important for us because we use hundreds and hundreds of liters of this every year. If we were using commercial media, this sort of project would simply not be feasible. Let me go back. Oh, I'm sorry, my video is not working. But the cells we click on the video, please. Go back one. There we go. So that's what the cardiomyocytes look like. They form these beating sheets of cells. They are commonly around about 80 to 90% pure. We have a couple of tricks to be able to take them up to nearly 100% pure cardiomyocytes. This system, which we published a few years ago, is also being commercialized. So you can buy this as a kit from Life Technologies. And these cells, it's been very clear that the utility of these cells is specifically very powerful for known genetic variants associated with electrophysiology. So that was the low-hanging fruit in the field. You can see that the first diseases to really be looked at with these iPS cell cardiomyocytes were long QT, and then there was some more kind of slightly more involved projects looking at things like hypertrophic and dilated cardiomyopathy. But our question has always been, it's all very well and good looking at a disease phenotype or a known genetic variant that causes a phenotype, but could we use this the other way around? Could we use the iPS cell model to essentially discover the genetic basis of a specific drug response? In this case, we're very interested in the adverse response to chemotherapy and whether or not these cells can essentially validate. So the first question is, do the iPS cells recapitulate a patient's phenotype? That's a very big question. And can we develop a suitable assay? A lot of these things have never been done with iPS cell cardiomyocytes before. So the first drug we chose to look at was doxorubicin, a very, very common chemotherapy drug, arguably the most common chemotherapy drug, used in about 60% of pediatric cancer patients. So these are patients who have no other alternative drug. For the breast cancer patients, it's used in triple negative and late stage patients. Again, some with the most poorest outcomes. It's very, very commonly used. Nearly 1 million patients receive this every year. And it has a well-established, almost right from the first discovery of this drug more than 50 years ago, that it causes cardiotoxicity. The number we throw out is between about 8 and 10% of patients will receive some level of toxicity. It depends on the dose. Breast cancer patients, the dose of doxorubicin they receive is limited by the cardiotoxicity, so not by the efficacy of the chemotherapy on the cancer, but actually by the toxicity. So for us, that means that if we could eliminate the toxicity or predict the toxicity, the patients could essentially have a higher dose, improving the efficacy of their chemotherapy treatment. Useful for us from a biochemical standpoint is that when the patients receive a dose of doxorubicin, you can detect troponin, one of the major cardiomyocyte proteins, in the periphery within about a week. So that tells us that cardiomyocytes die, giving us our first primary output for the in vitro assays. So this is all published. I will kind of skip through this a little bit, but essentially we took patients who had had doxorubicin, no toxicity, and then patients who had had doxorubicin and with toxicity, made iPS cells, made cardiomyocytes, and then tested them with a battery of assays. These are our patients. Oh, this didn't come out so well. But you can see the bottom 4 there from their starting ejection fraction, all kind of high and healthy, reducing to, in some cases, as low as 10%. If you have an ejection fraction of 10%, you're in real trouble. So, as I say, this is published, so I can just explain that For patients who had previously had toxicity, when we made their iPS cells, they were considerably more sensitive to doxorubicin than patients who had had doxorubicin and not had toxicity. And here's just a visualization of exactly what that looks by eye. So when we're doing these in vitro assays, without any kind of fancy assessment, just by eye, you can tell that these cells are more sensitive to the drug. We did a lot of drilling down into the mechanism, and to cut a long story short, we showed that, much to my disappointment, that actually many different pathways are different between the iPS cells from patients that had toxicity to those that didn't have toxicity. So that includes reactive oxygen species production. You can see it's considerably higher. DNA damage at lower doses of doxorubicin, sarcomeric alignment, and an interesting phenotype to do with mitochondrial dysfunction. So that's all very well and good. It seems to show that the iPS cell model does recapitulate a specific phenotype, which is what we were hoping for. But this didn't tell us much about the genetics. What is the basis behind it? I will add, this is the first time anyone had ever validated that there is a genetic basis to this drug response. Before that, it had obviously been hypothesized. But when you have the iPS cells, you know the only thing you're carrying over is the genome. The cardiomyocytes you make, they're de novo, they're brand new cardiomyocytes, so you're not dealing with a legacy of the history of that patient's heart. So here's a slide that many people in my field show all the time. This is all of the SNP variants that have been detected with a specific quality cutoff, validating the connection between this SNP and a disease. So there isn't one of these for doxorubicin-induced toxicity, but there is one that's nearly as good. So our collaborators at the Canadian Pharmacogenomics Network performed a GWAS. They had over 400 patients, and they discovered this variant in this retinoic acid receptor gamma. It has a reasonable p-value. That's actually just below the cutoff that we consider a gold standard in the field. For those that are interested in this sort of thing, the minimum allelic frequency in the control patients is down below 0.01, whereas in the patients who had toxicity, it's up at about 0.3. So very common in patients that have toxicity. So to use this kind of data in the human iPS cell models is not quite as simple as it seems. So what we had to do was go back to the original GWAS population, pick specific patients that had this variant and no other variant that had ever been associated with cardiotoxicity, even though none of them have ever been validated. So everything that was found in the most recent study, we excluded patients who had that. Anything that's been ever found in literature, we excluded those as well, and picked patients, because we know their genotype, that specifically have just this variant and no other confounding factors. That was very, very complex. That is a recruitment nightmare to try and go back and specifically re-recruit 3 patients that have previously been recruited. This included our nurse practitioners driving hundreds of miles to go and take blood in high school bathrooms to be able to do this. I apologize to them for ever asking them to do it. I never knew it would be so hard. But we did recruit these patients. We confirmed their genotypes. We know their drug response. And this is a second study when we're talking about reproducibility. This is a completely independent study done by my new postdocs, independent from all my previous work. And to my relief, it does reveal the same phenotype, that patients who have this variant, we're going to call it S427L, they do have higher sensitivity to doxorubicin. And then we've now kind of turned these assays into high throughput. So these are done in 384 well, which means we can get really high ends for these and feel much more confident with the data. So we see the cells die more easily. They produce caspase 3,7 at lower doses, and they still have this reactive oxygen species phenotype. DNA damage at lower doses, as we previously saw. So we then asked, that's very interesting, but can we actually validate that it is this gene that's important? So using CRISPR, again, very easy these days, we perform knockouts and overexpression. And to cut a long story short, yes, we do see the same phenotype. I think the caspase shows it well. This is by the cells either having or overexpressing or not or not expressing this single gene, this is the difference we see in their sensitivity to doxorubicin. All of the other assays in a very similar way. We spent some time looking at the genetics behind it, and long story short, as expected, the pathways that are involved in doxorubicin response, there is some differential between the patients who have this specific variant. And then finally, the gold standard answer to this is, can we take a patient who had toxicity, perform a single SNP correction, and essentially remove their sensitivity to the drug? So again, lots of CRISPR, and this is exactly what happens. So here is the control patient in the black. This is the variant, exactly the same line, this is the line with the variant, and then this over here is the exact same line with just that single SNP corrected, and we can show that we move that toxicity right across. And the same for all of the other assays, which means this variant is right up at the top of the chain of the doxorubicin response. So we're very happy that that seems to be correlated and making some sense to us. Of course, we didn't leave it here, so for my final slide, I'll say, because we knew this was a retinoic acid receptor gamma, and my background's in developmental biology, and I thought there was probably some more interesting questions there, we assessed whether specific and potent retinoic acid receptor gamma agonists could stimulate the retinoic acid receptor gamma to essentially act as a cardioprotectant. And my point is not really working very well, but here's the control. This is black, so they're LD50. The dose at which 50% of the cells die is usually around 2 micromolar. When we add in this drug, CD1530, which is one of the most specific and potent RARG receptor agonists, we take that dose to 14 micromolar. If you remember right back to the start, the difference between breast cancer patients who get this limiting dose, which is 240 milligrams per meter squared, with a theoretical toxicity of 3%, and sarcoma patients who get 450, so 240 versus 450, that changes the toxicity by around about 18%. So we here are changing that by 7-fold using this simple drug. The great thing about this drug is it's already bioavailable, so we could dose some mice just by adding it to their water before we treated them with doxorubicin. And for those who are not interested in the echoes, you can see the raw data here. So this is the change in fractional shortening after doxorubicin, and for the mice that are dosed with the CD1530, it about halves. Intriguingly, using all-trans retinoic acid does not recapitulate that, so it is also related to the specificity and potency of this drug. So I'd just like to thank my group. We've been going for about 2.5 years at Northwestern. Kyle Yang, who is responsible for doing most of the work in the RARG project, and of course our funding, and to say that we're always open to new collaborations. Our interest is in the model, so we're happy to take on new questions. And we'll do questions at the end in the panel. Thank you.
Great, thanks, Paul.